Hey, you are watching AliAziz YouTube channel.
Immunohistochemistry, IHC, methodology. AliAzizQualityEducation YouTube channel provided you the medical related topics videos. So don't forget the subscription button. Immunohistochemistry, IHC, is a powerful laboratory technique that marries the specificity of immunology with the intricate architecture of histology. By utilizing antibodies to detect specific antigens in tissue sections, IHC provides critical insights into the molecular underpinnings of diseases, particularly cancers. This article delves into the IHC methodology, exploring its principles, procedures, and the human elements that make it indispensable in modern pathology. The principle of immunohistochemistry. At its core, IHC leverages the highly specific binding affinity between an antibody and an antigen. Antibodies are proteins produced by the immune system that can recognize and bind to unique molecular structures, antigens, with high specificity. In IHC, these antibodies are used as probes to detect the presence and localization of specific proteins within tissue sections. Key steps in IHC methodology. The IHC process comprises several key steps, each requiring meticulous attention to detail to ensure accurate and reliable results. Tissue preparation. The first step involves obtaining a tissue sample, typically through biopsy or surgical excision. The tissue is then fixed, commonly in formalin, to preserve its structure and antigens. It is subsequently embedded in paraffin to create a solid block that can be sliced into thin sections. Sectioning and mounting. Thin sections of the paraffin-embedded tissue are cut using a microtome and mounted on two glass slides. These sections are then deparaffinised and rehydrated through a series of xylene and alcohol washes, preparing them for staining. Antigen retrieval. Formalin fixation can mask antigenic sites, so antigen retrieval is often necessary to unmask these sites and enhance antibody binding. This step typically involves heating the tissue sections in a buffer solution, which helps break the crosslinks formed during fixation. Blocking nonspecific binding. To prevent nonspecific binding of antibodies, tissue sections are incubated with a blocking solution. This step is crucial for reducing background staining and improving the specificity of the antibody-antigen interaction. Primary antibody incubation. The sections are then incubated with a primary antibody specific to the target antigen. The primary antibody binds to its corresponding antigen if present in the tissue. Secondary antibody incubation. After washing away unbound primary antibodies, the sections are incubated with a secondary antibody. This antibody recognizes and binds to the primary antibody. The secondary antibody is typically conjugated to an enzyme or a fluorescent dye, which allows for visualization. Detection and visualization. If the secondary antibody is enzyme-linked, a chromogenic substrate is added, producing a color change at the site of the antigen. Alternatively, if a fluorescent dye is used, the sections are examined under a fluorescence microscope. The resulting color or fluorescence indicates the presence and location of the target antigen. Counterstaining. To provide contrast and context, tissue sections are often counterstained with a general stain, for example, hematoxylin, which highlights the overall tissue architecture. Mounting and analysis. Finally, the stained sections are mounted with a cover slip and examined under a microscope. The patterns and intensity of staining are analyzed to draw conclusions about the presence and distribution of the target antigen. The human touch in immunohistochemistry IHC. While IHC is a highly technical process, the human element is paramount. The success of IHC relies on the expertise, precision, and judgment of the laboratory personnel involved. From selecting the appropriate antibodies and optimizing protocols to interpreting the complex patterns of staining, skilled pathologists and technicians play a crucial role. Their work requires a deep understanding of both the biological significance of the target antigens and the technical nuances of the staining process. Each tissue sample represents a unique case, often involving a patient awaiting a critical diagnosis. This awareness imbues the process with a profound sense of responsibility and dedication. Applications and impact. IHC has revolutionized diagnostic pathology, particularly in oncology. It is instrumental in cancer diagnosis. IHC helps identify the type and origin of cancer cells, guiding treatment decisions. For example, it can differentiate between primary and metastatic tumors. Prognostic indicators. The expression of certain proteins can provide prognostic information, helping predict disease outcomes and tailor treatments. Research and drug development. IHC is a valuable tool in research, aiding in the understanding of disease mechanisms and the development of targeted therapies. Conclusion. Immunohistochemistry, IHC, is a cornerstone of modern pathology, bridging the gap between molecular biology and diagnostic medicine. Its ability to visualize specific proteins within the complex architecture of tissues provides invaluable insights into disease processes. The meticulous methodology and the human expertise behind IHC ensure that each stained slide is not just a piece of art but a critical component in the journey towards accurate diagnosis and effective treatment. As science advances, the role of IHC continues to expand, offering ever more detailed and precise views into the molecular landscapes of health and disease.